So today I'm going to tell you how the technology has developed so that we can edit genes. One of the important uses for this is to do gene therapy, which means to somehow make corrections in our DNA, corrections that could solve some really nasty diseases. So really interesting stuff coming up. And at the heart of this is this new procedure called CRISPR. And so this appeared in Nature, which is probably the preeminent science journal in the world. And it had this story on CRISPR, the biggest game changer to hit biology since the polymerase chain reaction, but with huge potential pressing concerns. And of course, down here we have our potential designer baby, and we will touch at that in the end. Okay. So before we get into CRISPR and how it works and how you do gene editing, why do we need to do gene editing in the first place? Why are there mistakes in our DNA? So let's just talk a little bit about the molecular biology of mutations. So first of all, we have a lot of mutations, and mutations occur in our cells all the time. The principal reason there are mistakes, changes in the DNA code, is that every time the DNA gets replicated, every time a cell divides, you have to make a new strand of DNA. And this is done by an enzyme in a complex of other proteins. This enzyme is called the DNA polymerase. It makes the polymer. And what it does is it matches up C's with G's and T's with A's, etc. But at a very low rate, it makes a mistake. And the mistake in the DNA replication is on the order of 1 in a million times. Well, we've got 3 billion pairs of DNA, which would mean that every time we replicated our DNA, conceivably, we would make 3,000 mistakes. And these mistakes that do occur at a fairly high frequency. Not at that high, because most of them are corrected. Now, before we get into that, though, there are other ways in which we get mutations, changes in the DNA sequence. So about a third of them come from either chemical or environmental damage. So one of them is that oxygen, which we absolutely have to have for survival, is a very reactive molecule. And so oxygen can form things like peroxides, etc. And these can just be random chemical reactions that occur in our cells. If they occur, they can change the structure of a nucleotide. They can interfere with this polymerase reaction. So there's a certain level of that we just can't avoid. We can't depend on a highly reactive molecule like oxygen and yet avoid the potential damages. We can fix 99% of the problems that it causes, but not 100%. And then there are things like radiation, for example, potassium-40 exposure. Have any of you ever been exposed to potassium-40?
Probably.
What is the major ion inside your cells? In your blood, it's sodium, and in the inside, it's potassium. All right. We have a very high level of potassium in our bodies. So you've probably heard we have a little bit of carbon-14. We're naturally radioactive because of carbon-14. But the really big number of radioactive molecules we have are isotopes of potassium-40. So if you had a Geiger counter and you could count every radioactive event that's going on in your body, you could hear this thing clicking away. Potassium-14 would generate about 40,000 counts per second. All right, try and sleep at home tonight now after you that story. No way around it. Of course, X-rays. We're talking here a lot of it that comes in, manages to get through the atmosphere. Sunlight, of course, UV radiation, chemicals in our food and environment, etc. So it's just inevitable. Because it's inevitable, we have evolved ways to repair most of these mutations. So the cell has a whole set of different kinds of enzymes to repair different kinds of breaks, ways of recognizing. So for example, here, if a mistake is made and this G is paired with a T, that has put a mutation in at that site. However, because GT pairs are not canonical pairs, they will have a different chemical structure. That means that a protein can recognize this as, hey, this is not an ATP, this is an AC, an AT, it's not a CG, it's a bad base pair. And we have the enzymes that will actually go in, cut that DNA, remove the bad one, and put the good one back. And the other thing that happens is you can get breakage of your DNA. I mean, just 6 feet of DNA you have, it does break in the cells under normal conditions. There are enzymes that say, there's 2 ends of broken DNA. I'm going to put them back together and I'm going to hook them up. And so, for example, there have been some recent stories about looking at the Greenland shark. I don't know, this is an animal that can live for at least 200 plus years. And so when you sequence its DNA, does it tell you anything about how it can live so long? Well, it turns out that one of the differences is it has quite a few more genes devoted to DNA repair. So it's elaborated an extensive way of repairing these changes that we just can't avoid. Now, sort of stressing this because, well, before I get to that, but then the point here, I say very few mutations are harmful. Well, how can that be? Well, first of all, I mentioned that only 2% of your DNA codes for proteins. So most likely, these mutations are going to be in some of this repeated DNA regions that really doesn't make a functional enzyme. And so if we look in these regions, we find that, in fact, there's more variability in sequence, consistent with the fact that if you get a mutation there, it's not likely to affect your ability to reproduce. So we carry these along. All right. But in the case of, if it's in a protein coding region, I mentioned earlier that there are 6 different 3-letter combinations that code for some of the amino acids. And so if you change a 3-letter sequence that codes for valine into another 3-letter sequence that still codes for valine, it's not harmful. All right. So very few mutations are actually harmful and have an effect. But they do arise, and we're going to talk about some of them that cause serious diseases. Now, besides this, mutations drive evolution. They allow new variations to occur. So is a mutation ever actually beneficial? If any of us benefiting from something that happened in the last 5,000, 10,000 years? Mark?
Clearly. You're standing there being brilliant.
Are you calling me a mutant, Mark?
But in a good way.
Well, classic example, there's a mutation in the DNA that occurred probably in the last 6,000, 7,000, maybe a little bit that inactivated the mechanism that allowed us to catabolize, use as a nutrient, the lactose that we got from our mother's milk. I mean, that gene was only made, that gene produces the enzyme that allows us to use lactose, and it gets turned off when you're about 1 or 2 years old, because 1,000 years ago, people weren't drinking milk after that. However, there was random mutation, and for people who lived in that part of the world in which milk became a major food product, This was a big advantage if suddenly you could drink milk without throwing up.
Right.
And so probably about 80% of the people in the room are lactose tolerant. You're all mutants, all right? That's the example of a beneficial mutation.
On the oxidation damage, it seems like there's a fuzzy area between kind of natural, replication errors and the oxygen that surrounds us. Is this ozone or is this, how do we include it rather than just, part of the natural bio?
It's just natural oxygen. So natural oxygen is quite soluble. It's soluble in the water in our blood. It can react with H2O to produce peroxide. Peroxide is a very reactive molecule. We have these peroxidases in there specifically to convert it back to water, but you just got a low level of peroxide that you can't get rid of.
So unlike all the others, this is not something that will eliminate at the top.
No,
Okay.
right, no. Okay,
So this is free diatomic oxygen.
yeah,
Okay.
yeah.
Out of curiosity, we can't make vitamin C, and I can put some vitamin C in. Do you have any idea when we lost capability?
Well, so the question is, we can't make vitamin C, and why? So there are, in humans, there are about 50 things we can't make. I mean, one of the interesting things is we can't make half of the amino acids we need for proteins. All right. Why? Because it's a trade-off. One, if you could make all of the enzymes that you need to make those amino acids, but there's an energetic cost of that. To have all those genes and maintain them, and it's a complicated pathway. You don't make any of these enzymes or vitamin C with just one enzyme, the whole series. So it's evolution has driven the fact that it's more efficient for us not to bother with those genes. We will just eat other animals and get the amino acids directly from them. All right, and that's what a vitamin is. It's something that we don't make ourselves, but we can get it by eating other living creatures.
Isn't it not energy efficient to produce all that junk DNA? How come it's not getting?
All right. Oh, so why do we make all the junk DNA? And we don't really have a really good answer for that. And so the question is it ultimately going to be useful? Looks like it's not all useful. Again, it's a trade-off, and probably some of it gets eliminated, but we don't really understand that. All right. Okay, now I'm stressing this in part because the gene editing techniques that have been developed make big use of these enzymes that repair DNA. I mean, if you want to cut DNA or put DNA back together, etc., there's no way you're going to do this as a biologist the way a chemist would, put the reactions together and have a chemical reaction to form the molecule you want. The way to do it is the cells out there are already making enzymes that can put DNA together, that can insert nucleotide. I mean, among the dozens and dozens of just different kind of repair enzymes, etc., some that are very useful for this editing. So they're going to come up in that context. All right. So let's go to the CRISPR story. Fascinating story. And in a way, it starts with this guy, Francisco Mojica, who was a microbiologist. He was interested in kind of, the really strange microorganisms that can grow in extreme environments. He worked at a university in Spain, and very early on, I think as he had just finished his graduate work, he was working with a sort of very odd DNA sequence in E. coli. Now, bacteria generally don't have these long repeated sequences in their DNA the way we do, because sort of to your question, to make that DNA is an energetic cost. So they've eliminated most repeats. But in a few random bacteria, there was a stretch of DNA that had a repeat. So it was sort of like this. You'd have some piece of bacterial DNA, and then some odd fragment, and then another repeated piece of this bacterial DNA, and then some odd fragment. You might have 20, 30, 50 of these, just bacterial DNA, odd fragment, bacterial DNA, odd fragment, etc. And so he was looking at this sequence, and he just published a paper saying, I found this odd thing. It looks like repeated DNA in humans almost, but it's in this odd bacterium. He looked through the papers and found out there had been one other guy published something similar a few years before that, but other people responded to Mojica's paper, and so they started finding similar things in widely different kinds of bacteria. There were a bunch of reports, so they had to come up with a name for this, and so Mojica came up with this name. He called it CRISPR, and CRISPR stands for clustered regularly interspaced short palindromic repeats. All right. Nobody can remember this whole thing, but CRISPR works out pretty well. All right. You can kind of remember what CRISPR is. All right. So as Mojica kept working with this, and in 2003, so this was, about 5, 6, 7 years after he published his paper, there were more and more DNA sequences put it into the international databases. And so he just submitted the sequence of this repeated piece of DNA into the database and says, is this like anything else? And what popped back at him was it said that stretches of what you see are identical to the sequence that have been found in many different types of bacterial viruses. That in fact, what you've got here is a piece of bacterial DNA and then some fragment from some random virus, and then another piece of DNA, and then some fragment maybe from some different virus. And so he was a sharp guy, and he saw really quite immediately that this was important. So here's in the book described as, here's what he said. He said, I just discovered something really amazing. Bacteria have an immune system. They're able to remember what viruses attacked them in the past. He said this to his wife. In a few years, you'll see this thing that I just discovered will be written about in newspapers and in history books. And he was right. He was right. Okay, so need to know a little bit about viruses and DNA, viruses and bacterial cells. So just as there are viruses that attack us, there are animal viruses, there are plant viruses, but probably the most abundant biological structure in the entire world is the world of bacterial viruses. It's estimated that an ounce of seawater may contain 50 million bacterial viruses. More than anything else, we live in a world of viruses everywhere. And so here's a bacterial cell, and here are these bacterial viruses. They look a little bit like the lunar lander, right? All right. This is not a drawing. This is a real electron micrograph. And so these legs attach them to specific proteins on the surface of the bacterial cell. And then the DNA in these viruses, some have DNA, some have RNA, again, gets injected into the cell. And once that DNA gets in there, it's read into proteins, and the proteins start making all of the pieces of the virus. And then the viruses get assembled inside the cell. And then the last step is the proteins trigger the fact that the cell wall in the virus loses its integrity. The cells burst and they release hundreds of new viruses. All right. So there are many different kinds of viruses. This is a kind of a typical structure of one class, but I show this just to show you all the different shapes that viruses can take. So they're very tiny. A typical DNA molecule in a virus, whereas ours has 3 billion bases, a typical viral DNA might be on the order of 50,000 or 100,000. So, you just think of the fact that you need, roughly a couple thousand pieces of DNA to specify for a protein. So if you had 50,000, you could maybe pack the information for 25 proteins into that. And that's sort of a pretty accurate estimate of what's in a typical virus. Because if you look at these, there's just usually 2 or 3 different kinds of proteins that will make the head. There's some proteins that will make this injection part, proteins that make the legs. So you don't need that many different kinds of viruses to make proteins, rather, to make a virus. And of course, it's the interesting story of are these living organisms or not? I mean, most biologists would say no, but they're certainly self-replicating. I mean, they're just little AI machines. They got everything they need to make lots of them. So there's this constant war out there between viruses and bacteria, because the viruses will kill the bacteria. So it makes sense that an immune system to attack the virus would have evolved. Just a little side note here is, 2 side notes. One is that there has been a long area of research which continues to try to maybe use these bacterial viruses to fight bacterial infections. And this is getting a little more attention now because antibiotics have been very successful, but the bacteria have become resistant to the antibiotics. Antibiotics new ones are not being found at a high rate. So the idea of going back and finding particular viruses, and that would attack and kill disease-causing bacteria is an attractive hypothesis. And of course, it has its own problem. You're engineering a virus that can attack cells, and it all depends on exactly how specific it is.
So this is a little bit far out, but what if we had, we've got a series of substances that are made to produce the result of a living organism. But what if their product was something different, like a saw? You have a saw, and then you have a way that can be touched to receptors in other people. Does that make sense?
I'm not following you. We might have to pursue this later. Okay, wait.
Maybe, but I had to say something.
Yeah, okay, thank you. All right, so let's just summarize where this story stood when it was in the hands of the microbiologists. So what they knew generally was that a virus invades bacterial cells by injecting its DNA in. And what they had found with the CRISPR RNA is that the CRISPR RNA was composed of little pieces of cut up bacterial viruses. And these little pieces of viral DNA was then organized into a piece of DNA in the bacterial genome, such that you had these bacterially produced spacers, and then a piece of viral DNA, another spacer, a piece of viral DNA. So literally a kind of history of the various viruses that had attacked this bacteria in the past. And somehow, if a piece of DNA got injected into this cell from an invading virus, and it was a match to something that had been seen before, it activated a mechanism that allowed it to recognize this as a virus and go to the virus DNA and cut it up. So they kind of had an idea of what was going on. That's why they said it was like an immune system. But how any of this worked was a complete mystery. All right, so Mojica wrote up his results, and he had real trouble getting anybody to publish it. For one thing, he was talking about an immune system and extreme bacteria. who cares, right? This is kind of exotic, odd organisms he was talking about. He got rejected by a couple of journals, and it took him 2 years to get his paper published. I think got published in 2005. Now, other microbiologists were interested in this. And there was a microbiologist at UC Berkeley, a woman named Jillian Banfield. And they were thinking, well, there's some sort of gene silencing event occurring on here. And the hot topic of the time was called RNA interference. That's another really interesting way of silencing genes. It really kind of falls up under the topic of epigenetics, and I didn't really touch it last time. But they thought there's some RNA interference mechanism that underlies how this immune system works. So this woman microbiologist at Berkeley went to her computer and typed in, who knows something about RNA interference? And one of the experts that popped up was another Berkeley professor, whom she really hadn't met before. And this Berkeley professor was Jennifer Doudna, who turns out to be one of the heroes of our story. So this kind of jumping ahead is biography, not so much of Jennifer Doudna as of the whole CRISPR story, written by Walter Isaacson, came out 5-6 years ago. Man, it has a copy of it there. How many of you have read this book? Anybody read this book? Okay, a handful of people have read this book. Very interesting. I really like this book. Very different in a way in that Isaacson really went into the details of the people, especially who involved in working this all out, really showed a lot of the complexity, the interpersonal reactions, the politics, the money, all of this aspect behind what was a major scientific discovery. So anyway, the two Berkeley professors got together for lunch and said, all right, let's work on this as a collaboration. And as more and more work started to be done, there were enough people working around the world doing some work on this, maybe 50 laboratories around the world. They decided we need to get together every once in a while, and we need to have a conference. So they had the first CRISPR conference was in 2008 at Berkeley. All right. Now it was, and they were making pretty slow progress to figuring out what was going on. So just 3 years after that, Jennifer Doudna went to another science meeting. This is something that scientists spend a lot of time doing. To you, the taxpayer, it looks like, why are they going to Puerto Rico, every other year for science? Well, I don't know. All right. But these conferences are really important. So Jennifer Doudna went to a conference in Puerto Rico, and she had just a chance meeting with a scientist from Sweden, who is actually French, named Emmanuel Charpentier. And let's see, now, I guess, yeah, at this point, I was thinking Scott and Beatrice have usually come. Yeah, I should call him. Who here speaks French? What's the proper way to say Charpentier? It's like, yeah, all right, because I'll butcher this woman's name. So she had also been working on CRISPR, and she had added something important to the knowledge. She found out that there was another RNA molecule that was essential for the CRISPR immunity situation to work. And Charpentier didn't have any real expertise in working with DNA, especially kind of structural studies that she wanted to do. And so they decided over lunch that they would have a collaboration. Now, one of the key things to know about how science works in the modern world is the view might be that Doudna and Charpentier went back to their labs and got out their test tubes and started working. No, that's not how it's done. They went back to their labs and they say, I've got to find somebody to do this work. All right. So I need a postdoc who's going to do this. So they each found a postdoctoral worker in their lab who was interested in the project. And so here was the team. So there's Emmanuelle Charpentier, Jennifer Doudna. Her postdoc was Martin Jenick, and Charpentier's postdoc was Christoph Shilinsky. Now, Isaacson, in writing this book, makes a really interesting point about this. So these four became the core of this collaboration. And so he has this in the book. He says, the collaboration was like a model United Nations, a Berkeley professor from Hawaii, her postdoc from the Czech Republic, a Parisian professor working in Sweden, and her Polish-born postdoc working in Vienna. Because Charpentier had been in Vienna, left the postdoc, moved to another lab in Sweden. But these four people were all working on the project. And especially the two guys here, they were really doing the work. And it was very interesting in a way in that Vienna and Berkeley are 10 hours apart. And so he describes how the two young postdocs would twice a day, they would get on Skype and say, all right, this is what I did today, and this is how my results got up. And the guy in Vienna said, all right, well, I'll follow up on that. And then the next morning, the Vienna guy said, well, last night, this is what I found out. All right, this is what you should do. They were in this 24-hour cycle of back and forth and back and forth. I mean, and the two, lab leaders, they definitely participated in this whole conversation. I don't want to in any way not say that they were the leaders behind this, but it was a fascinating collaboration. So it became this 24-hour project. Now, in the end, of course, this all results in the Nobel Prize in Chemistry in 2020 for these two people, and I think they deserved it because they were in it from the beginning to the end. But the book, I think, is wonderful in the way Isaacson really gives the other people their due and their moments of fame in the book. All right. So, what is CRISPR? What did they figure out? Okay, so the essence of it, and we aren't going to go through the chemical history of how they arrived at this, but here's how it works in the end. One of this DNA sequence has these fragments of viral DNA in it. And this DNA sequence also has regular genes that encode for enzymes. And the most important enzyme in there was this one that was called Cas9, CRISPR-associated gene number 9. So this gene encodes for a big protein. This protein is a binding site for an RNA molecule called the guide RNA, which is part of it sort of is made by the bacterium. It's a part normal bacteria DNA. And the RNA and the protein get together to form a complex. And the function of this complex is twofold. First of all, it holds a little piece of the viral DNA, so it can go looking for viral genomes that match this viral DNA. That's its one function. And the second one is the protein, when it finds and binds to a viral genome, will cut the viral DNA. So it has a targeting function. Let's find a viral genome that matches our little piece of viral DNA. And it has the function to destroy that viral genome by cutting it. So the structure is such that here's the scaffolding part of the RNA, and then you attach to it, and in the cells, in bacterial cells, there will be a whole bunch of these made with different fragments here. And then this is sort of just bouncing around in the cell, if you will. And if it bangs against a piece of viral DNA through this combination, this process of DNA hybridization, it can say, ah, there's an exact match. And this would be the piece of the virus DNA here. And now it's attached itself to this little fragment carried by the bacteria. And once this whole complex assembles, then the protein has a mechanism within it that does the chemical reaction that cleaves the DNA. And so this is the viral genome that has now been cut open. All right, questions about that part, because that's kind of critical.
Does the DNA and the protein stick together?
Yes,
So one recognition, the other one cuts.
so once the recognition, the protein actually binds that viral DNA in just the right position so it can cut it.
So
And an interesting thing is the place it's very precise, and the precision comes about from the fact that the protein is carrying this fragment to position it. And so this part here is the targeting sequence carried by the bacteria. This is from the viral genome.
two quick questions. One, the inserted DNA, I assume is mostly junk DNA. I mean,
The which DNA?
so we cut out the virus, we cut out the objectionable DNA, and we've inserted new DNA. And I'm assuming the new DNA has no function.
Those fragments, you mean?
Yeah.
The main thing is that they're quite small, so they would be much too small to function in, yeah.
Then the other question is, in a way, the bacteria has a gene sequencer in it. It has this database of all these viral fragments. It can match them up. And I'm wondering if that was an inspiration for actual, I mean, you can see it almost as an inspiration, like for the nanopore technology stuff. Have a database like a UC database, find fragments that match up. In this case, we're throwing them away. In the other case, we're making awesome identify. I'm just wondering if any of your heroes of biochemistry use CRISPR as not only a Nobel Prize winning thing, but also an inspiration to how they sequence genes.
Not really, because the way gene sequence it's really done in a very different way.
Yeah.
They really couldn't get any lessons.
It's kind of cool. It does have a skill that recognize stuff.
Yeah,
I'm sort of wondering about the form of that diagram.
yeah.
Is that right? If you look at it under a microscope, does it look like that, or is it more simple?
So it's very diagrammatic in that these shapes, of course, will be much more structured, and the protein will look quite different. So no, this should be looked at as just a vague sketch of where the pieces are, but the actual atomic level detail would be quite a bit different. It has now been worked out in an atomic level detail, but
Because sometimes those diagrams are really interesting.
yeah. I mean, for example, just the question of does it sit on the face of it or is it inside of it? And the answer is a mix of both. No, okay.
There are little scissors.
Then, yeah. Oh, the little scissors are real. Oh, yeah. No, they're not.
How is this initiated?
So basically, once you would get a match, what would happen is, so you want, once you get this bind, actually, I can just show you this. So like this, if this is the viral DNA, you've got these little fragments here. Now, my diagram isn't accurate in that each of these fragments are going to be carried by the CRISPR, but they're all bouncing around here. And at some point, they're going to line up exactly. You're going to have this lock and key thing where they're going to lock up where the complementary sequence is. And once that happens, that will change the shape of the whole complex and set off the cutting reaction.
But beyond that, who starts it?
Well,
Automatic?
oh, so the CRISPR with these fragments, there's going to be literally thousands of these molecules produced all the time, basically just bouncing around there, waiting for a virus to invade. Just as in our bodies, we have we make antibodies to things that we aren't exposed to right now, but we were exposed to measles or measles vaccine in our youth, and we still have those antibodies circulating in case a measles virus comes in. And so this is kind of the analogy of that. In these bacterial cells, they have these CRISPR molecules just bouncing around there in case a virus comes in.
The big green there is the viral DNA, right?
Right.
And then, so does it get cut in lots of different places, like, or does it get just cut in one place?
No. So as I'm sort of trying to show my little cartoon, it turns out there's only one place where there's a match. There are lots of fragments sort of bouncing around and interacting, but there will just be one place.
Is that basically kill it? Is that inactivate? Is that how it's really spread?
Yes, it does. So, and this especially is likely to work with viruses because viruses are so compact. They don't have this extra sort of non-coding DNA. So there could be times where it could come in a place that did not inactivate the virus. That could and probably does happen, but at a very high frequency, it'll inactivate some essential gene required for the replication of the virus. Mark.
So I'm trying to think, if this stuff is bouncing around inside the cell and it's looking randomly, it will attach because there's a match. How fast is that going on? I mean, like, is this a thing that you have to go wait for 2 weeks, or does it happen?
So you said, how fast is this going on?
Yeah.
All right. So this is, I think, one of the most important numbers in science, and nobody knows this number. Biochemists know this. The bottom line answer to your question, Mark, is if this is an enzyme,
Yes.
and you had, let me just, as a different example, let's say you have an enzyme, and its function is to bind a glucose molecule and then cut it in half. If it's just sitting there and glucose molecules are bouncing around, how frequently is this enzyme just sitting there likely to be hit in the right spot by a glucose molecule? And the answer is that at the level that if glucose is present in the cell at a sort of medium concentration, it'll hit that 1 million times per second.
Oh.
So I mean, that's the other thing that's so hard to grasp in terms of, but that's what drives out, how can this chemistry work? Just by random collisions. There are a lot of random collisions. It's just another world of time that occurs at the level of chemicals. And we have some enzymes, particularly the ones that attack these peroxidase molecules that get formed and can be damaging. These peroxidases approach catalytic perfection, meaning they can catalyze almost 1 million reactions per second. All right, because whenever one of those hits, bang, they got to split that molecule. Yeah, so this random collision thing is hard to get in your head, but it drives the whole thing. All right.
Questions kind of